Hi, I'm Shyam. I'm in the 11th grade at the Illinois Math and Science Academy in Aurora, and my internship this summer was tracking Zebrafish Cilia utilizing Mathematica under Dr. James Choi. So here's a basic introduction about the zebrafish, a freshwater fish. It's native to Southeast Asia, and it's a very common aquarium fish as well. It's been commonly studied by biologists around the world, and the cilium of this fish, and I quote here, is a previously little studied cell surface, and it's emerged as an important organelle in vertebrate cells. And to further study this organelle, um, we we had this zebrafish data, and we tracked it frame by frame, tracked each cilia as they went through time. Cilia. Let's first open up the cilia and see how they look. So here we have one of the images of the cilia. This is in what Mathematica terms called a manipulate, and what a manipulate is, you can change the settings, right? So here we can change the scene right here. We'll change the second here, and basically here we're just importing all of the TIFF files and selecting number 55, which was determined as the best test case scenario. Number 55 here, it's this diagonal thing, and in the middle here it's the brightness per thing, right? It's per image. So this is image 1 all the way to image 350 something, 60 something, and this is the maximum brightness in the image, right? The maximum brightness in the image. And here we have a formula that almost like it's like a linear fit. Here it's not linear, but it's a fit. Here's logarithmic, actually, a logarithmic fit to this pattern here. And to analyze these images, the basic image analyzer up here was created. This is the original unedited TIFF file, and on the right here, what we did is we divided each data point in here by this value, right? Given the image frame. So we plug in the image frame here, we get a value here, and we divide all the points in here by the max point to get this really crisp, clean, like, you can't tell it by eye, but it's much better to work with in Mathematica than this one here. Mathematica works a lot better with this version than this one. So, we, again, as seen here, we determined to use TIFF file 55. It was very clear, it had clear cilia, and it had a very unblurred view compared to some of the other ones. So right here we are rotating the image right here. As you can see, we're rotating the image so that we don't have any of this other data around here, right? So we have a straight image. It really works well in Mathematica that to have the straight image, and also is pleasing to the eye. Then we just crop off the edges so that Mathematica functions do not have to worry about this data out here, this image out here, and it can focus sincerely on the cilia themselves. So let's crop the bottom off, and here we go. We have this crop, and let me move the changes back real fast. But yes, so we had this crop that just focused solely on the cilia. This was found right here, and again we have one of these manipulates where you can go through each of the frames and see how the cilia move over time. Now what's going to happen is we're going to use two different functions: image feature track and image corresponding points, to try to see if we can track these cilia over time. So the first one we use is image feature track. It's more precise. It gives a more accurate result, but again less quantity with that quality. So we use image feature track here, and we get this 3D view, of the tracking almost, right? So this axis is over time. So this is the time axis over the frames, and this, as you can see, is just x and y, right? Y right here, x here. You see it, y, x, and over time here. So as we can see on the xy plane, this is all in one spot. It's tracking a feature really, really well, and over time we see that it's going up a little bit, around, I would say, 60, 70 frames from the first frame. And in this way, we can track the cilia. And image feature track, Mathematica found four. Let me open this up. Four different feature tracks. 1, 2, 3, and 4. My favorite by far is this one because it really shows this almost. It almost resembles a sine curve, as we see here. And again, we will interpolate this line later on. But for now, image feature track. Mathematica is able to track a cilia, right? Since really it's tracking a feature, right? It's tracking a feature in the image. But since each feature almost corresponds to a single cilia, which means that this line follows the path of a single cilia. And like I said before, it was able to track 4 different cilia. The same feat, the same achievement was attempted by the second function, image corresponding points. So we use image corresponding points. The syntax is all here. Image corresponding points on the data, the TIFF file. And here, what we're doing here is we're clustering the points, right? So image corresponding points is a much lower threshold than image feature track, and it really jumps around here a little bit. So what we're trying to do is we're trying to cluster the points into a group, right? Into a group of points all in the same location, right? So we can do this 3 different ways. First, using the distance formula, right? So points that are around near to each other using the distance formula. Then we can group by x coordinate. And we can also group by y coordinate. And we do that here. This is by the distance formula, the x coordinate, the y coordinate, and then we cluster 2 more times, just to be more precise, just to group them all in, a bunch them in together further and further. Then we extract the groups of the corresponding points right here, and like we saw here, we just we view each line. And as you can see here, again, we have this similar thing to what we saw before. A feature, right? A corresponding point being tracked all the way from the bottom to the top, right? Through time, through the cilias, it's being tracked. But here we see it's much longer than what we saw before, right here, right? This is the same scale. Over here it's much shorter. Over here it's much longer. And this is what I'm talking about. Where the corresponding points has much more quantity. It's longer, and they find more, right? So image corresponding points found 7, but the quality isn't so good, right? So there might be jumps between here. It's not consecutive, right? With image feature track, it's all consecutive. So basically, just tracing up the box, it's just like before, except a bit less quality, but a lot more quantity. So now the next task is to interpolate this data to create a more flowing line through the images tracking the cilia, right? So we can more easily see this line without having to look at specific points. Next, a 3D model of all the cilia through time. So basically, I am extracting these cilia and just looking at them, just looking at the cilia with an orange-yellow color. You'll see that in a minute. And so we just put this all together. So we put in the interpolation right here. We put in the image feature track lines. We put in the image corresponding points lines. We put in the 3D image, the 3D model of the cilia. We put in this is just a 2D vision right here, and all we have this really nice analyzer that's able to track points, right? So this analyzer almost you can choose whether to see image feature track or not. Boom, boom. You can see whether to see image corresponding points or not, and you're able to choose which line to see, right? So again, my favorite is line 3. Keep that there. You're able to see all of them as well, but again, my favorite is line 3. Let's keep that in here. We're able to see the points in the image, right? So the points as I go, as you can see, image feature track, the red one, has far better accuracy, right? So that all the points are clustered together, but image corresponding points, much more quantity, right? They're more farther apart. You're able to see more, But again, the accuracy might be a bit less because they're so spread apart that much. But that's fixed while we interpolate the points. As you can see, we have this blue interpolation line that goes through them, and you have the option of adding in the 3D image. We'll try that right now. Let's see if this works. It's going to take a while, but this should work. If it doesn't crash. Yes, there we go. So we can see this 3D image of the cilia layered on top. As we can see, there's this like line that just travels the edge of the cilia, and it's tracking the points. Okay, let's get rid of this. It takes up a lot of memory on my computer, this 3D image. So we have that, and down here we can see the points as they go through. So this one's for the red, right? We see where it's tracking, where this feature is at, and this one is for the green right here. We see where this feature is located at. And in conclusion, by utilizing these two functions, this project was successfully able to track cilia through time and through images. Image feature track found a more accurate, precise tracking of the cilia with less quantity but more quality, while image corresponding points, on the other hand, was able to track with significantly more quantity, as we see here, more quantity but far less quality, spanning a range of far more images, but with cilia being tracked further apart. By combining this with interpolation, this project was successfully able to track zebrafish cilia. Now, at the end of this, I'd like to thank Dr. James Choi for providing me with the opportunity. I'd like to thank Dr. Saxena from UIC for providing this data and really giving us the opportunity to track these zebrafish cilias and expose us to this really interesting field, right? Of this field that I've never looked at, and how Mathematica can help us attack this, right? Attack this tracking of zebrafish. Thank you very much. This was a very fun, very educational, and very great project. Thanks. Thank you.